All we really need to do now is get some live bacteria. Oh, Iran, perfect. Put them over there. Is everything okay? Great. Hi. So here we are at the bench. And today we're going to actually pour agar plates and see just why the agar that Angelina introduced into Koch's lab is so special. So I've asked Michael to join us and help us pour plates. Here we have the broth and here we have the agar. And as you can see, we're going to add the agar to the broth and put it on a hot plate and subsequently pour plates. The agar is boiled, we put it aside, it's cooled, and now we're going to be actually pouring plates. The plates are poured one by one, and then subsequently we're going to leave all of the plates cool and set so we can then inoculate with bacteria. The plates have cooled, everything seems fine, and all we have to do at this point is inoculate them with bacteria. Bacteria are fine, and here, Michael, come a minute, there seems to be a problem. This culture is fine, whereas this culture seems rather mysterious. It looks as if the bacteria have been killed or lysed. Michael, why don't you take the bacteria and prepare a series of dilutions of both, and we'll check it out. Now, in the meantime, let me show you what agar is all about. Here's the plate. I've taken a spatula, and I'm just going to carefully detach it from the plate and show you the kind of jelly that Angelina prepared for Walter. Here we are, we're inoculating plates, and Michael is doing it systematically, one by one. We've prepared a series of dilutions from the first culture, culture A, which has the good bacteria. And then we made a similar series of dilutions of culture B, the mysterious culture. Now, for each plate, bacteria are added, and then on a turntable, we spread the bacteria over the surface of the agar. Once that's finished, the plate is turned over and then subsequently labeled with that dilution that we used for that particular culture. In this case, B-1. And here we are. We go on through the series. We've completed the series of culture A, and we're just about to complete the series of culture B. Afterwards, because we still want to try to understand and examine what is the nature of the mysterious culture, I'll ask Michael to prepare a lawn, a plate with a continuous culture of bacteria, and then take from the series of diluted B and add drops one by one onto the surface of the bacteria and see, do the drops have any material, any toxin, any killing substance that actually kills the cells, as we've seen. Tomorrow morning, we'll come by and see the results and try to understand what has happened. Thank you, Michael. We've now inoculated a plate, and it has a continuous surface of bacteria growing. And to these bacteria, we're going to add drops of the mysterious material at various dilutions, as you can see here. So carefully, a single drop of 5 microliters is added. It'll be absorbed, and if there's anything there that's killing the bacteria, we should see the effect. So 24 hours have passed. The plates have been incubated at 37 degrees, and I asked Michael to give me a set of representative results. And the winner is the highest concentration of bacteria that were plated. This is the 10 to the -1. And as you can see, there's a continuous lawn of bacteria that cover the entire surface of the plate. For comparison, I'm showing you a plate that we did not inoculate, so you can clearly see the difference between bacteria growing on the plate and a plate that has no bacteria at all. The next concentration was the same bacteria of culture A, but diluted 1,000 times, 10 to the minus 3. And as you can see, we can see numerous colonies of bacteria. Think about it. Each colony represents one bacterium that fortuitously landed on the plate. It grew, divided, until there were about 1 billion bacteria in one particular colony. These are bacterial clones. And you can see that the number reflects the dilution. Because as we go to the next dilution, 10 times more diluted, we can see that in 10,000 times diluted, we have clones. About 10 times less than what we saw in the previous plate. So as you can see, we were able to culture bacteria on agar plates and actually count individual bacterial clones. Now the question is what is in the mysterious material? So we plated it, and at its highest concentration, nothing. No bacteria grew. So there was something in that culture that lysed and killed the bacteria, some toxin. And that is what we have to try to figure out, if it can directly kill bacteria growing on a lawn. And for that, as you recall, in the last experiment, we plated a lawn of bacteria and then added 4 concentrations of the mysterious material onto the bacteria. Now, as you can see, in the highest concentrations, there's total killing. But as we go to higher and higher dilutions, less and less of the material, we don't see a gradual killing, but rather discrete small spots, which we call plaques. We've discovered viruses. And these are the viruses that have killed the bacterium. They're discrete. These are plaque-forming units. And as you can see, as we dilute them tenfold, we can actually quantify the number of phages, or viruses that kill bacteria, in that mysterious solution. I don't quite understand. Somehow it got contaminated. Who knows?